Have you ever wondered, does God exist? Have you ever looked at the world around you and thought to yourself, was this created or evolved? Are rocks really millions of years old? Did dinosaurs roam the world not too long ago? Is there evidence for a worldwide global flood? Is the Bible reliable? Is it accurate? Is it true? Join us now for this episode of The Evidence.
Good evening and welcome to The Evidence. This is night number 2. If tonight is your first night, don't worry. Good News TV is recording this series and they have DVDs available for those, or will have DVDs available for those who missed night number 1. This is a 6-part series that goes through some of the big questions that people ask all over the world and down through time. Questions such as, does God exist? Where did life on planet Earth come from? Is there evidence for a global, worldwide, catastrophic flood, just as the Bible mentions? Does archeology prove the Bible, fact or fiction? Is there evidence for the existence of God in science and archeology? The evidence looks at these big life questions through the lens of science and archeology. Now, in our first presentation, we discovered that our universe is expanding in every direction. Einstein concluded this, so did Alexander Friedman, so did George Lemaitre, so did Edwin Hubble. In every direction, galaxies, stars, solar systems are slowly drifting apart. This indicated that our universe, if you go back in time, must have started at some particular point in time. That the universe itself must not be eternal. There has to be a beginning. Many scientists have concluded that that beginning was a Big Bang. However, we looked at 7 major problems, and there are many more that are being discovered through scientific discoveries today that show that the Big Bang model is beginning to fall apart and is not a sufficient model to describe everything that we're beginning to discover today. One of those things that we have looked at is that the universe spins in both directions. Moons spinning, orbiting planets in both directions. There's whole galaxies that are orbiting or that are spinning in the wrong direction. We also looked at a star, the Methuselah star, which is older than the universe itself, at just over 14 billion years old. Then we concluded last night's presentation by looking at the second law of thermodynamics, which states 2 things. Number 1, that the second law of thermodynamics says that usable energy is converted to unusable energy irreversibly. So in other words, if I had a hot cup of coffee and I set it right here on the stage, that hot cup of coffee would eventually grow to room temperature. And it would stay at room temperature and would not go back to a boiling cup of coffee at any point in time unless you put a stove under it or something to heat it up again. So usable energy is transferring irreversibly to unusable energy. Thus, we can conclude, if our universe was eternal, there should be no usable energy left in our universe. The stars would have burned out by now if the universe was eternal. Because we still have burning stars, because we have a sun that rises every morning, because there's still energy being expanded or used in the universe, we can conclude that our universe must have had a beginning. A second thing that the second law of thermodynamics states is that the elements or the things in our universe gradually move from order towards chaos, irreversibly, never from chaos to order. If we lived in an eternal universe, we would not see any remnants of order in our universe. Everything would be chaos. But since we do see order in our universe, we must not live in a universe that's eternal, because an eternal universe would have swept everything into chaos. according to the second law of thermodynamics. Thus, according to the evidence we studied last night, we have to have lived, or at least the probability of us living in a universe that had a beginning is very, very high, higher than the probability of living in a universe that's eternal. We also discovered that things that have a beginning logically must have been created by something that existed before it. Like a chair. If a chair has a beginning, there must have been something or someone that made it. If the universe had a beginning, there must be something or someone that made it. Tonight, we're going to move past galaxies and past stars, past solar systems, right down here to our own blue planet. We're going to be looking at how life formed on planet Earth, the evidence for whether life formed through a random process or whether life was created, designed, and purposely put here on planet Earth. We begin with the story of Charles Darwin and how the theory of evolution came into being. Now, at the age of 22, Charles Darwin got the opportunity of a lifetime, a round-the-world trip on a ship called the HMS Beagle. He was 2 months at sea, and during these 2 months at sea, he discovered and began to learn things that shook the world of science like nothing else had. Once he got on the ship, he was handed a book called Principles in Geology, written by a geologist by the name of Charles Lyell. Lyell's theory said that rocks were millions of years old. Canyons took vast amounts of time to form. All of this Lyell called deep time. Darwin had 2 months to read, and by the time the ship had landed in South America, he was fully convinced that Lyell was right, that everything that he saw in geology, whether it was a canyon or rock formation or whatever he saw, could be explained through Lyell's deep time, or millions of years. There was one other influential book that Darwin read on his voyage. It was a book written by somebody he was related to, Erasmus Darwin, his grandfather. Erasmus Darwin had written a book named Zoonomia. Zoonomia brought together the idea that every form of life on planet Earth was related to one another. So all life forms on planet Earth were related to one another. As the ship began to travel around South America, charting the coastline, Darwin sought to explain everything that he saw through these 2 books that he had read that deeply impacted him and shaped his ideas. This proved pivotal when the boat finally landed among the Galapagos Islands. There, Darwin noticed slight changes in mockingbirds that were on the islands and mockingbirds that were on the mainland. Now, this was revolutionary for Darwin because up until this point, not only Darwin, but Charles Lyell, who wrote Principles in Geology, and culture at that time believed that species were fixed or couldn't change, that once a mockingbird with all of its features had become a mockingbird, that's all it would ever be, that none of those features would change. So when Charles Darwin saw the features of the mockingbirds on the Galapagos Islands were slightly different than the features of the mockingbirds on mainland, he began to rethink this popular idea. Finally, after the Galapagos Islands, Darwin made his way home. And once he got home, he began to formulate his theory of evolution. And this is how that formulation went. From reading Charles Lyell's book, his theory said over vast amounts of time, from his observations on the Galapagos Islands, small and slight changes within species. From his grandfather's book, Erasmus Darwin explains how all species are connected, and now comes the novel or new thought through a common ancestor. Darwin had completed his theory of explaining how the variety of life on planet Earth could have arisen through natural processes unrelated to a supernatural being. His theory was known, or came to be known, as the theory of evolution, the book that he wrote, The Origin of Species. In Darwin's book, the illustration of an evolutionary tree showed how all living things, plants, fish, animals, humans, all trace their roots back to a single life form, one primordial cell that started it all, and it began to evolve into something more and more complex from that point. Darwin's theory was an attempt at explaining the entire world and the diversity we see in it from one original life source. This is what he wrote. All organic beings which have ever lived on this earth have descended from some what? Read it with me. One primordial form. One primordial form. What does that mean? Well, how everything evolved from a single cell. How there was a single cell and began to split and become more and more complex. With this idea, Darwin had a theory that explained life on Earth, but there was one major problem. How did this first life, this one primordial form, come into existence? If God didn't create the first life, then where did it come from? Darwin sought to explain everything through natural means. So in Darwin's mind, the first life had to come from some non-living stuff that existed before it. Rocks, dirt, chemical elements, non-living matter. The big problem with Darwin's theory is that in order for Darwin's theory to really work, be valid, function, you have to explain how that first living cell came from the non-living matter that existed before it. If you cannot explain, or if it is seen to be impossible for a living organism to come from non-living matter or stuff, for a cell to come from rocks, if that is proven to be impossible, then Darwin's theory breaks apart or breaks down. The entire theory of evolution is based on moving from non-living inorganic substances to that first living cell and onward from there. So how did the first life come from non-living matter? That's the real question. This is how Darwin puts it in his own words. Origin of Species, page 159. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would what? Absolutely break down. So, what we're looking for to either prove or disprove evolution is there evidence that through successive slight modifications you can get from a rock to a cell, or from inorganic substances to an organic living molecule, an organic living organism, a single-celled organism? Is there evidence for that? Now, the origin of life from non-living matter is called chemical evolution or abiogenesis. The evolution of life or living organisms from inorganic or inanimate substances. Now, theories about this, about abiogenesis or how living things come from non-living things, trace all the way back to ancient Greek philosophy and Aristotle. Aristotle was one of the first individuals who mentioned that it could be readily observed that aphids, the tiny insect aphids, came from raindrops. Rotting food is where flies came from. Hay bales produced mice. And rotting logs, well, everybody knows that if you have a rotting log, you're probably going to get an alligator. The theory of Aristotle, thank goodness, was later disproved by a man by the name of Antony Leeuwenhoek in 1680. Antony Leeuwenhoek disproved his theory because he was the one who first discovered the microscope and was able to see that flies actually lay eggs. Now, with technological advances in science, by the 1800s, scientists thought that life easily could have arisen from a single cell. Now, the views of single-celled organisms in the 1800s was basically that a single cell was a globule of plasm, and all you needed to explain was how that globule of plasm developed a cell wall. Or, in simple terms, how a dollop of Jell-O develops a cell wall to contain it. How do you contain this dollop of Jell-O? Once you explain that, you've got your first living cell. That was the understanding of scientists in the 1800s. Today, we know that a single cell is far, far, far more complex than a dollop of Jell-O. Would you agree? Absolutely. In order to have a living cell, you need literally hundreds of different functioning proteins, all working together. Proteins are the very basis of living cells. Without proteins, and without hundreds of different types of proteins all working together, you cannot, will not, ever have a living, functioning cell. And you won't have the beginning of Darwin's tree. Now, Darwin must have understood that proteins were the key to understanding the first life, because in a letter dated to February 1871, Darwin said to a friend that the first protein must have been formed in a warm little pond with all sorts of ammonia, phosphoric salts, light, heat, and electricity present, that a protein compound was chemically formed ready to undergo still more complex changes. So just to review, before you can have a single cell, what do you need? Proteins. Before you can have a single cell, what do you need? Proteins. Without proteins, you cannot have a single cell. And without that single cell, you cannot have life or the theory of evolution. So everything depends on what? Proteins, the formation of proteins. So now's where it gets a little bit complex. We're going to be talking about protein formation. So hang on with me. We'll get through it together. Now, proteins are three-dimensional structures. Think of a hammer, a saw, and a screwdriver. Those are three-dimensional structures as well. They have different sides to it. You can pick it up. You can use it for a specific task. And proteins are like that. They're three-dimensional structures that are formed into a specific shape to perform a specific task inside the cell. Some proteins provide the structure of the cell, like a skeleton or a cell wall. Other proteins speed up reactions in the cell to not just thousands, but millions of times faster than that reaction would occur were that protein not present. Those proteins that speed up reactions are called enzymes. We're going to be talking a little bit more about enzymes later. But enzymes are, you could think of it kind of like this. If you need to cut down a tree, which would cut down the tree faster, a hammer or a saw? A saw. What's the difference between a hammer and a saw? The shape. And that's what enzymes are. Enzymes are proteins that are in just the right shape to fit in a particular process in a cell to speed it up to millions of times faster than it would occur if that enzyme were not present. Other proteins are used to transmit signals inside, outside the cell. Other proteins are used to transport waste and nutrients in and out of the cell. Others act like an army, binding to foreign invaders. How many of you feel like you're in Biology 101? Yeah, but this is important stuff to understand. So proteins are critical, and you need all these different types of proteins all functioning together. So how is a protein formed? Well, proteins are made up of things called amino acids. And a protein is a long chain of amino acids, and that chain is twisted and bent and folded until it's in a specific shape. And that's the specific shape that it will hold and function in the cell. So proteins are a chain of amino acids. Amino acids come in 20 different types. So there's 20 different types of amino acids. You've heard of 31 flavor ice cream, 20 different types. You have the Rocky Road, the vanilla, the chocolate, 20 different types. With a protein, those different amino acids have to be lined up exactly in a precise sequence. So to illustrate this, on the screen, I have amino acids represented by colored blocks. So if you were to form a functioning protein, you would have to form a functioning protein with an exact sequence of amino acids. And it might look something like this: a pink amino acid followed by a blue one, followed by an orange one, green, then blue, then pink, then purple, then orange, and then finally green. And once that green amino acid completes that specific chain, now you have a functioning protein. And you don't have a functioning protein if even one of those blocks is missing or in the wrong order. Does that make sense? So this is how proteins are formed. Amino acids bind together in a very specific sequence. Now, some people may connect a little bit better with numbers. So let's say that the amino acids were represented by numbers. If you have 20 available amino acids, you can view them as a sequence of numbers. So you have amino acid number 1. And in this protein, it has to be followed by amino acid number what? 5. Not 4, not 3, not 2, but 5 and only 5. And then it has to be followed by amino acid number 20, and so on. You have 4, 5, 1, 16, and onward. If you have one single amino acid missing or put in the wrong place, the whole protein chain will fall limp and will not function in the cell. Just a single amino acid in the chain, and the whole thing goes limp. Now, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids lined up in this protein. How many amino acids need to be lined up to make a small protein? Well, an average protein is 300 plus amino acids. But that's an average one. The largest one, the largest one is much bigger. We'll get to that in just a moment. So in order for chemical evolution to work, in order for Darwin's theory to work, you need to form proteins from amino acids. And those amino acids must be able to form from chemicals. Okay, so these are the steps that you need for evolution to work. You need rocks, chemicals, dirt, elements, non-living stuff to form amino acids. Then you need those amino acids to line up together and form a protein. Then you need a bunch of proteins to get together and form a cell. Then you need that cell to divide and do its evolution thing and form horses and ducks and cows and bees and beavers, right? So that is the process. Rocks, amino acids, proteins, cells, beavers. Got it? That's the process. So we're right back here with amino acids. You have to be able to form amino acids from chemicals or rocks, inorganic substances. Amino acids need to bind together in some way to form a chain if you're going to have a protein. So there has to be something, whether it's magnetic or chemical bonding or some sort of bonding that brings them together. Amino acids need to line up in a very specific sequence. You need 1 followed by 5, followed by 20, followed by 4. You need that specific sequence or the whole protein won't work. Once a correctly sequenced chain is formed, then it needs to be folded into a 3-dimensional structure, like a hammer or a saw or a screwdriver. It has to be folded into a very specific shape. You need a whole bunch of these proteins all correctly folded to make a living cell, and now you've started where Darwin's theory starts, the first living cell and the theory of evolution. So Stanley Miller in 1953 attempted to simulate Darwin's warm little pond where the first amino acids were supposed to have been formed. So he wanted to really start there at the beginning and form amino acids from chemicals. So he combined methane, ammonia, hydrogen, water, and he put it all in an anaerobic or oxygen-less, oxygen-deprived environment, a vacuum. Okay, so there's no oxygen in this contraption. And you'll see here, I don't know if you can see my laser light, but here's where he put all of the chemicals, and then he put a heat source underneath it, boiled it, turned it into a vapor. The vapor rose through the tube and eventually entered into this chamber, and he simulated lightning strikes in that chamber with electrical impulses, boom, 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 boom. And as the vapor was being pulsed with this electricity, it began to condense and reform into a liquid, and down here is where the results would be collected, down in this P-trap, down at the bottom. After 2 weeks of doing this, zapping these chemicals over and over and over, amazingly, he was able to produce 13 of the 20 different amino acids. Now, mind you, this is a perfect environment. Could this happen in the wild without oxygen? Could this happen in the wild? We don't know. But in a perfect environment, 13 of the 20 amino acids needed for a protein were formed. And it was hailed a scientific breakthrough. But did this close the case on how proteins are formed? Not yet. Miller had accomplished the first and easiest of all steps. The more challenging problem was to get those amino acids to line up with one another in the correct sequence. None of them did. None of those amino acids formed proteins in that little P-trap down there. The only thing he was able to do is form separate amino acids, 13 of the 20 amino acids down there. So amino acids could form if the conditions were perfect. But what about in the wild where the conditions aren't perfect? Dean Kenyon, the author of Biochemical Predestination, says that in his book, life might have been biochemically predestined by the properties of attraction that exist between its chemical parts. In other words, in the wild, it just happened. Amino acids just lined up on their own. They lined up and sequenced themselves correctly. It was biochemical predestination. One of Dean Kenyon's students challenged him on this point and stood up and said, Dr. Kenyon, the only thing we observe in nature, in protein synthesis or production, is that proteins are formed through the information in DNA, the DNA in cells. Without DNA, there's not a single protein that's formed on planet, not a single protein that's observed to have been formed on planet Earth. You have to have DNA if you're going to have amino acids lining up correctly. And without DNA, it's impossible. You're just left with random chance without DNA. It's like trying to come up with the right sequence by guessing the numbers in a combination lock. If you have to sequence a protein chain with the right sequence of amino acids, it's random chance. Now, on a lock, on a combination lock, there are 3 or 4 spaces. Here on this combination lock, we have 3 spaces. 1, 2, 3. In each space, there's 10 numbers to choose from, 0 through 9. However, in sequencing a protein, you don't have 3 or 4 spaces. You have 300 to 400 spaces. And in each space, you have 20 available amino acids, not 10, to choose from. So what's the odds of having that size of combination lock cracked by chance? We're going to look at those odds in just a moment. So let's look at the odds of a protein forming by chance. Now, Doug Axe, he's a molecular biologist, did this calculation. By the way, the largest protein on planet Earth is Titan. This protein looks like this. It's 34,350 amino acids long. That's how long the amino acid chain is. 34,350 little amino acids. That's huge. Now, if you had a protein 1 amino acid long, you would have a 1 in 20 chance of getting that protein right? Just 1 amino acid long, you have a 1 in 20 chance because you have 20 available amino acids. You have a 1 in 20 chance. Stanley Miller showed that 13 amino acids could form in just 2 weeks. What are the odds of a protein that is not 300 amino acids long, but a small one that's only 150 amino acids long? What are the odds of that protein being formed by chance? Random chance. If you have a 1 in 20 chance of forming a protein that's only 1 amino acid long, what about one that's 150 amino acids long? That's 150 slots and 20 different options at each slot. You ready? That's the number. 1 times 10 to the 195th power. I can't even wrap my mind around that number. That's huge. Now, just to put that into perspective, in the whole known universe, scientists estimate 10 to the 80th power of elementary particles. That's protons, neutrons, and electrons. There's a greater chance for you to find 1 marked proton or 1 specific electron searching the entire universe than for 1 simple protein chain to be lined up correctly by chance. Now, let's say that for every second on a watch, a new amino acid combination was tried. Every second. Every single second in the primordial soup, a new combination of amino acids was tried. According to probability, you would need 1 times 10 to the 195th seconds. Now, if the universe is 14 billion years old, you would have a total of approximately 4 times 10 to the 17th seconds in the total universe since the Big Bang, supposed Big Bang, until now. You don't have, in other words, you don't have enough seconds available in the entire universe to form a single small protein by chance. There's not enough time in the entire universe, even if you had 14 billion years, to form a single small protein that's only 150 amino acids long. How big is the largest protein? 34,000 amino acids long. We don't need a single protein for a cell, for a working cell. You need 150, 200 different proteins for a single cell to work. So you don't have to just do this once. You need to do this again and again and again and again. So, how do you form a protein by chance? Well, amino acids must form from chemicals. Amino acids need to be sequenced correctly. And we've seen that there's not enough time in the entire universe for that to happen by chance. Amino acids need to form correct bonds. And here's what the amino acids look like. Amino acids form a specific bond of peptides called a peptide bond. And without that bond, amino acids won't line up together. They have to have this bond called a peptide bond. The problem is that these bonds occur only 50% of the time. Half the amino acids bond together and the other half don't. At least not with a peptide bond. So the chances of all the bonds being the right bond, a peptide bond, are 1 times 10 to the 49th power. Not only do you need the right bonds, but you need the right bends. Amino acids come in 2 types, just like people, right-handed and left-handed. 50% of amino acids are right hand, 50% are left hand. The problem is that proteins only use left-handed amino acids. So, the odds of getting only left-handed amino acids are again 1 times 10 to the 49th power. So amino acids forming in the correct sequence, the odds are 1 times 10 to the 195th power. Amino acids forming the correct bonds, 1 times 10 to the 49th power. Amino acids only being left-handed, 1 times 10 to the 49th power. In the unlikely possibility that all these things could happen and a single protein does form, you still haven't arrived at the place where Darwin began, because it takes 250 to 400 proteins to form a simple single cell. So let me ask you, do you think that you could have a single-celled organism formed by chance? What do you think? Do you think the odds are in favor of that happening? Absolutely not. There's no way. And many scientists around the world who understand this stuff much better than I do are coming to the conclusion that if you're going to have a single-celled organism, the most crucial thing you need is DNA. Without DNA, there's no way that you can have life on planet Earth, because DNA is critical for protein formation. Without DNA, you can't have proteins forming, because what DNA does is it tells the cell exactly where the amino acids need to line up to form a specific strand of DNA, and exactly how to fold it into what exact shape. Without the information in DNA, you cannot have life. And without life, you cannot have a theory called evolution. The whole thing breaks down. So, really, it all comes down to the information in DNA. How did the information get in the strands of DNA in everybody's cells here? How did that information get there? Because without that information in your body right here, right now, there would not be life. Wouldn't exist. You wouldn't exist. Explaining how information got in DNA is a real challenge for evolutionary biologists. So how does this all happen? Let me show you how proteins are formed in a simple cell. This is a cell right here, right there. If we zoom in to the cell nucleus, right here, that's the very center of the cell, you'll find that it's filled with a tightly wound scroll called DNA with all this information. And there's the DNA, you see it there, and it's all wound up inside the cell nucleus. When a protein is needed by the cell sends a machine called RNA polymerase into the cell nucleus, and it goes along the strand of DNA until it finds the information that is needed to build that specific protein. It's incredible how this happens. So RNA polymerase goes along, he's searching through the library there in the center of the cell, boom, he finds the right book, pulls out the book, and opens it up. And as he unwinds the strand of DNA, he begins to read it. He puts markers on the two ends, protein markers. By the way, RNA polymerase is a protein machine. And it begins to run along the DNA strand and make an exact copy of the information in the DNA. So he copies the book. He says, I'm going to copy the recipe for making this particular protein. Boom, got the recipe. And that recipe comes out as something called RNA. RNA then travels out of the cell nucleus into the middle of the cell. RNA travels to another protein machine, and this protein machine is called a ribosome. The ribosome is going to read the instructions or the recipe to form a protein. So the ribosome reads the instructions from the RNA, and it says, hey, these are the different amino acids I need. And it calls for the amino acids, and the amino acids are brought by another enzyme, protein enzyme called tRNA. And it brings all the correct amino acids needed to form that protein, and the ribosome sequences that protein chain together from the information that came from the RNA. The recipe just follows the recipe. And out comes a protein chain. Now, the protein chain is not functional yet. It still has to be folded into a specific shape. So it enters another protein machine called a chaperonin, and that's a barrel-shaped machine inside of the cell. There inside that barrel-shaped machine, that protein chain is folded over and twisted and folded and twisted and folded and twisted until it's in the exact shape that it needs to be in to function in the cell, and only then is that protein released from the chaperonin to function in the cell. And by the way, once it's released, it's shepherded by another protein machine that brings that protein to the exact place in the cell where it's needed. Without the information in DNA, there's no way that you would have life. The problem comes down to how did that information get in there? There's the protein chain, there's the folded protein. Now, you can't have truly functional DNA without enzymes. The RNA polymerase that reads the DNA, that little machine that goes into the library and finds the right book and opens the page number to the right page and makes a copy of it, that's called an enzyme. It's just the right shape. Enzymes help speed up cellular activity to unimaginable speeds, 1 × 10 to the 18th power. Enzymes are specific tools made to speed up the job. So the same job without an enzyme would take literally millions of years to perform without that enzyme. So it can't be performed. Enzymes are necessary to read the information in DNA. However, DNA is necessary to make enzymes. Let me say that one more time. Enzymes are necessary in order to read the information in DNA. But the information in DNA is necessary in order to make enzymes. Maybe you didn't get it. Proteins are needed if you're going to get the information out of the DNA. However, you need the information in the DNA in order to form the first protein. Which one came first? Was it the protein or was it the DNA? Because you need the protein in order to read the information in the DNA, and you need the information in the DNA to form the first protein. Which one came first? The only plausible explanation that I can come up with, and maybe you can come up with a better one, is that they came into existence at the same time for that purpose. Because if they didn't come into existence at the same time, neither one could exist or be usable. You need both proteins and DNA in order to have each other, or in order for each other to function or work. Without DNA, you cannot have life. So if there's one thing you need to take away from this seminar, it's that DNA is necessary for life on planet Earth. And without DNA, you can't have life. And by the way, DNA is in every living organism on the planet. If it has a cell, it has DNA. Whether it's bacteria, or whether it's a fish in the sea, or a bird in the air, or whether it's you sitting here tonight, everything that has life has DNA, and without DNA, it cannot have life. Now, scientists are realizing this, that you cannot have life without DNA. You have to have the instruction codes or the information in DNA for life to begin. Bill Gates said that human DNA is like a computer program, but far, far, far more advanced than any we've ever created. Bernhard Olaf Küppers, a German biochemist, had this to say. He said, the problem of the origin of life is clearly basically the equivalent to the problem of the origin of biological information. In other words, our query or our quest to discover where did life on planet Earth come from, all comes back to where did the information in DNA come from. If you can answer where the information in DNA came from, then you can answer where life on planet Earth came from. Without information, you can't have life. So let's look at the information in DNA. There needs to be, number one, for there to be information in DNA, there needs to be a language. Spanish, English, Chinese, or in DNA's case, it's a 4-letter code, AGCT. If you were speaking computer language, it would be a binary code, 0 and 1, 0, 1, 0, 1, 1, 1, 1, 1, 0, 0, 0, 0, right? Well, what sort of information can you make with just 4 letters? Just so you realize what sort of information you can create with just 4 letters, think about how much information you can create with just two numbers, 0, 1, 0, 1. The entire world's information is stored on just zeros and ones in computers. Now you've just doubled that, more than doubled that. And they're discovering now that with this 4-letter code, you can have multiple layers of information parallel to each other in the same code. I mean, it's just almost unimaginable how much information can be stored in these tiny DNA structures. So you need language. Then there needs to be a communication channel. If you know a language but you don't have a mouth, you can't communicate that language. For DNA, it has to be able to transmit that information into RNA. Without RNA coming out of the cell, that information is trapped down in DNA. Third, the information needs to be meaningful. If you're wanting to make a protein and what you're getting out of the DNA is a recipe to make spaghetti, it's not useful information, is it? Well, it may be useful to make spaghetti, but it's not what you need. So you need information that corresponds to life. You need meaningful information that's going to produce the proteins necessary for life to form. So it has to be meaningful information. DNA's code has to be specifically sequenced, and it has to have specific information. So where did the information come from? First, the information in DNA has nothing to do with the actual physical DNA structure. And let me illustrate this for you. I have two pieces of paper that I'm going to present on the screen. One has information and one does not, but they both are made of the same stuff. Ink and paper. Both are made of the same material, but which one has information? Is there one that has information and one that doesn't? This one, the English Renaissance of the arts, this one, just ink and paper. Just because DNA is made up of a specific chemical parts has nothing to do with the information that's coded in it. Information and the physical parts are not synonymous. You'll never be able to explain how the paper and ink in a book chemically evolved and then use that chemical evolution to explain how Shakespeare was written. Right? Information cannot be explained by chemical evolution. Information on planet Earth has only one known source: intelligent beings. There's no other known source for information. Let me illustrate this. You're familiar with the SETI program, the Search for Extraterrestrial Intelligence. This is NASA's program that scans outer space looking for signals, codes, anything that smells like an alien. Now, what would happen if scientists suddenly picked up a code from outer space? Beep, 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 beep. SOS, SOS. What would they conclude? Hey, there may be intelligence out there after all. Why would they conclude that? Because every scientist infers that information only has one source, and that's an intelligent source. So if you're receiving information like an SOS code from outer space, even if you haven't seen the alien, even if you haven't received a package in the mail, if you're receiving information, it must come from an intelligent source. And isn't that what we all infer when we're, let's say, hiking through the desert and we see petroglyphs on rocks? I don't think there's a person here that would automatically assume that those rocks must have been formed by millions of raindrops over millions of years pounding on those rocks, producing those pictures. No way! We would automatically assume, wow, there must have been an Indian civilization that lived right here. There must have been an intelligent source that produced this, because this looks like, smells like, has the appearance of information, and information only has one source: intelligence. We naturally infer when we see petroglyphs, this must have come from an intelligent source. DNA contains a more advanced information than anything we have ever built, designed, or made. That information in DNA that is absolutely necessary for a working cell to exist and for any organism to have life came and had to have come from an intelligent source. The DNA book had to have been written by an intelligent author. The masterpiece of DNA was sculpted through the working of some creator. When you look at DNA, the DNA molecule, you aren't just looking at some ink and paper. You're looking at Shakespeare. You're looking at something that came from the hands of something that's intelligent. It was designed. Henry Quasler, molecular biologist, says that the creation of new information is habitually associated with conscious activity. Richard Dawkins, evolutionary scientist, even said that the machine code of a gene is uncannily computer-like apart from differences in jargon. The pages of a molecular biology journal might be interchanged with those of a computer engineering journal. And Dr. Crick, Francis Crick, Nobel laureate DNA research, said that biologists must constantly keep in mind that what they see was not designed, but evolved. Now, why do you have to always keep that in mind? Because what you're looking at indicates that it was designed. And so you have to continually tell yourself, why do you have to continually tell yourself that? Why can't you just say, no, this looks designed, it follows logic, maybe it was designed. The only reason that you would have to not consider that route is if you're protecting some standard scientific model. If you have to protect a standard scientific model, then you cannot consider anything that exists outside of that model. Now, what about junk DNA? Only 2% of the genome of DNA is used to make protein. So scientists have assumed that 98% of our DNA must then be junk DNA. Time magazine put this out September 6, 2012. They say, but it turns out they were wrong. In an impressive series of more than 30 papers published in several journals, including Nature Genome Research, Genome Biology, Science and Cell, scientists now report that these vast stretches of seeming junk DNA are actually the seat of crucial gene-controlling activity. Junk DNA? Nope, it's essential for you and I to have life. Can't just snip it out and get rid of it. We need all of it. Now, the next crushing blow came with the discovery of genetic entropy. What's genetic entropy? Genetic entropy says that genetic information degrades over time. This supports devolution rather than evolution. Your genes are degrading as you get older and older. The information is gradually crumbling apart at the seams. Every time a cell divides, 3 mutations occur. These are the mutations that cause us to get old and die. A portion of the mutations in each person is passed on to their offspring, so every generation begins with more mutations in their DNA than their parents. So rather than evolving, we're actually degrading. And because of genetic entropy, unless there's some sort of discovery or some sort of intervention in the human race, we will continue to have our genome crumble apart. This is following the second law of thermodynamics, isn't it? Everything moves from order towards chaos. Brianna Pobiner, anthropologist, says that mutations are basically the raw material on which evolution acts. So in order to have evolution, you need to have mutations. If one creature is going to evolve into another, it happens through the mutation of the genetic code in DNA. However, all the evidence in science shows that mutations can only remove or alter information that is already present. Mutations can never add, can never add new information. It can only remove information that was there or alter the information that's there. It can't insert new information. This loss of information can sometimes have beneficial side effects, such as in sickle cell anemia. Sickle cell anemia is a disease that affects individuals whose blood cells are mutated to be moon-shaped rather than the regular donut shape. And it allows them to be immune to malaria and diseases like this in Africa. However, the mutations also have a host of other problems that many people with sickle cell struggle with, causing them to be more prone to bacterial infections, blockages, and even a higher risk of stroke and death. Mutations work in the wrong direction for evolution by destroying information, not creating new information. Thus, even mutations are a problem. Now remember, Darwin said, if it could be demonstrated that any complex organ existed which could not have possibly been formed by numerous successive slight modifications, my theory would what? Would absolutely break down. So here's Darwin's dilemma. You need proteins if you're going to have life. Stanley Miller was able to produce amino acids from chemicals, but there isn't enough time in the universe for the amino acids to line up in the right sequence for even a single small protein. There's no way that you can get proteins by chance. This problem is compounded because you also need the right bonds and also the right bends. And then you need to fold the protein, and then you need to duplicate that process 250 to 400 times. You cannot get a single cell. You cannot get proteins through chance. You have to have DNA in order to get functioning proteins. DNA is needed to form proteins and enzymes, both of which are needed to read the information in DNA. You need DNA for proteins, and you need proteins to read the information in DNA, so you can't have one without the other. It is the information in DNA that is crucial to making proteins. This information cannot be explained by chemical evolution, and we readily observe that the information comes only from intelligent sources. Junk DNA is a myth. Our DNA is degrading with every generation. And with this information, we can conclude that life on planet Earth could not have come through a random or undirected process, such as evolution claims. Life on planet Earth, the evidence points to intelligent design. That at some point in time, the first gene, the first strand of DNA was coated with information to produce proteins that would produce life on planet Earth. Friends, I think that we've seen for a second night in a row that there's evidence for a supernatural being, an intelligent being, a being who produced life on planet Earth. Thank you for joining us for tonight's presentation. Tomorrow night, Darwin's Dilemma. You aren't going to want to miss it. Thank you again, and have a good night.